ONL Therapeutics is an emerging biotech company dedicated to protecting the vision of patients with retinal disease. We are developing first-in-class inhibitors of fast signaling, which has been shown to be relevant in a wide variety of retinal diseases, and ONL 1204 is our lead candidate for retinal detachment with an IND planned for next year. And we've recently completed a $5 million financing, which included our existing investors as well as new investors, including Novartis, which will fund our IND-enabling program as well as allow us to continue our research into additional indications. Before I get too far, I just want to say how lucky I am and what an honor it is to work with such an enormously talented management team and world-class scientific advisory board, many of whom you'll recognize. And before I talk about the development program, I'd like to tell you a little bit about why we think that targeting fast biology is so important. Fast signaling is activated by the fast ligand binding to the receptor at the cell's surface. This activation leads to two distinct signaling pathways. Most people are familiar with FAS's role in death signaling, but we believe that FAS's role in immune response and inflammatory signaling may be equally or even more important in retinal disease. It is this signaling which amplifies the signal, which recruits more immune cells with ligand to further activate and amplify the disease and the tissue damage. And it's been shown to be activated in a wide range of diseases. ONL's FAS inhibitors block both these types of signals, and we have demonstrated protection of multiple cell types in a wide spectrum of disease models. Here, my colleague Dave Zaks and his team at the University of Michigan demonstrate the ability of our peptide MET12 to protect photoreceptors in a model of retinal detachment. The same group earlier this year showed that this peptide not only protects RPE cells in a model of dry AMD, but it also prevents the activation of microglial cells, which we believe is an important feature of disease progression. Jiang et al. At Louisville showed that this peptide also protects from inflammatory damage in a model of non-infectious uveitis. And at the end of my talk, if you'll excuse me, I'm going to talk just a little bit about our early work in glaucoma. So let me update you on the ONL 1204 development program for retinal detachment. First question is, why retinal detachment? And the most important answer is because there is a continuing and significant unmet medical need. In spite of the wonderful surgical techniques that are performed by folks in this room on a daily basis, the average patient with a macula-off retinal detachment affecting central vision, even with successful surgery, will lose 3 to 5 lines of best-corrected vision. So clearly there's an unmet need, and we believe that if we inject patients with 1204 at the time of diagnosis, we can protect those cells until you do the surgery. It is also a very unique clinical development opportunity. This is an acute anatomic event. As such, it's much more straightforward to develop preclinical models that have a similar anatomic feature. The biological driver of vision loss in this indication is well established, and it's our target FAS. It also provides an opportunity for rapid readout. With that single injection at diagnosis, vision after retinal detachment stabilizes in about 6 months. So in 6 months, we'll be able to get a readout on the efficacy of our new molecule. And it's pretty rare in our business that we get to give a single treatment and have a readout in 6 months. And finally, because it is a surgical trial, we will have vitreous tissue readily available to us from the vitrectomies that will enable us to do molecular biomarker work, which we don't get to do in ophthalmology very often. So here we show that ONL 1204 is really an exquisite inhibitor of photoreceptor cell death in our rat retinal detachment model. And with a little love and a whole lot of effort led by Jana van de Goor, we now have a product candidate with really superb pharmaceutical properties. And we are excited to get this into the clinic. So we're on track to file an IND in the middle of next year with a financing transaction of some sort to fund those clinical trials. As I said, we recently closed our Series A along with an NEI grant that provides us the funding to complete the IND-enabling studies, as well as further our research into other indications. With that, I'd like to digress just a little bit off the retina to talk a little bit about our early work in acute glaucoma, because we think this work demonstrates the potential for fast inhibition in more chronic inflammation-related diseases of the eye. This is work that we're doing in collaboration with a group here at Scapins, Meredith Gregory Cassander, and it's really been a terrific collaboration. The model here is the bead model as described by Chen et al., and microbeads are injected into the anterior chamber. They then clog up the trabecular meshwork, and you get an elevation of IOP. There are two important pieces of data on this slide. The first is that IOP peaks at 3 days and then is sustained for about a month. And the second is that ONL 1204 does absolutely nothing to lower IOP. So in the first experiment, we used 2 different doses of ONL 1204, and then we went out, and these doses were injected at day 0 along with the microbeads. And as you can see, we went out 28 days, and we looked at axon density and retinal ganglion cell density, and you can see that both doses were protective, and you might see that there's maybe a dose response, although it wasn't powered for one. But then we asked the question, can we treat this in a more clinically relevant way? Most patients don't show up on day 0, they show up with elevated IOP. So we did the experiment again. This time we injected the drug on day 7, and if you remember, IOP peaks at day 3. And what you will notice is that the protection is just as good as it was with the day 0 injection. And we think this is pretty exciting. What I'm not showing you is not only are we protecting these important cells, but we're also preventing the upregulation of inflammatory markers like IL-1 beta. So we think that there's great potential for applying our fast inhibition technology to a wide range of ocular diseases. And with that, I'd like to thank you, thank the organizers, and I look forward to discussions throughout the day.